Welcome to the CIBM Center for Biomedical Imaging Breakfast Science Seminar number 12. It's my great pleasure to introduce you to Jean-Philippe Tirant, who's going to be chairing this session as the CIBM Signal Processing EPFL CHUV section head. So thank you, Jean-Philippe.
Thank you very much, Pina, for an introduction. Hello, everyone. Welcome indeed to this Breakfast in Science seminar. It's a great pleasure first to see so many of you, not really to see, but I would love to see you. But anyway, to have so many of you attending, I'm sure we'll have a fantastic Breakfast in Science seminar today, because I am extremely happy to welcome and introduce my very good colleague, Professor Inti Zlobec. Let me very briefly introduce Inti. So Inti received her PhD in pathology in experimental medicine from McGill in Montreal, Quebec. And already at that time, she combined two main interests on her side, pathology and mathematics, to develop biostatistical models for predicting the response of patients to radiotherapy in the specific case of colorectal cancer. And also she proposed an optimized workflow and optimized workflows for biomarker analysis. Then after that, she moved to Switzerland, first to Basel, where she was a privat docent. And then she moved to Bern, where she's an associate professor at the University of Bern. And she's the leader of the Translational Research Unit and the Tissue Bank Bern, that's the name, which are two core facilities for biobanking, tissue-based visualization, digital workflow. She also works intensively on image analysis and on the exploitation of next generation so-called tissue microarrays. I'm sure you will hear about microarrays today. So she leads the digital pathology group, which first, on the one hand, implements digital pathology in routine diagnostics, which is a very important revolution in the domain. And clearly, Inti is one of the most recognized leaders in implementing and developing digital pathology, so implementing digital tools in the clinical workflow, where there is a huge need for that kind of revolution. And on the other hand, she conducts very advanced research on image analysis, histopathology image of colorectal cancer, to go towards automatic diagnosis to refine molecular subtypes and prognosis. She is the president and co-founder of the Swiss Consortium for Digital Pathology, which aims at promoting this discipline. And we are very, very lucky to have, in the context of the CIBM, we have very good collaboration with Inti. We co-supervise also together with Bezad, one PhD student, Christian Abbé, and we collaborate with several postdocs and researchers. Some of them are present today in the context of machine learning for digital pathology. So really, it's very important, and I'm very happy to have Inti today. She's really a recognized leader in this domain of really changing the world of digital pathology, from microscope to digital tools. And I'm excited to listen to your talk today and to familiarize the CIBM community to your fantastic work. So Inti, thank you very much for accepting our invitation. And I think the floor is yours.
Philippe, thank you so much for this really wonderful introduction. I'm really honored to be here and to tell you something about digital pathology. So I'm just going to share my screen, and I hope that you can see my screen perfectly. Okay. So again, thank you very, very much for the invitation to present to you today a modality which maybe you're not totally familiar with, and that's digital pathology. And we are sort of in this third revolution, which I'd like to present to you today, but you'll certainly see that we are probably way behind. And it's known that we are way behind radiology in the sense of working with digital images and working on AI in digital images. So I'd like to cover a couple of topics. First, of course, is what is digital pathology, and then go over some clinical applications, namely some basic tasks and also some advanced tasks that are presently being worked on, and also some research examples. And in this context, I'll also show you what Jean-Philippe was mentioning, namely these NGTMAs or tissue microarrays that we work with very, very often in the lab. So if you see this picture, something looks a little strange. So this is in fact our microscope auditorium here at the Institute of Pathology in Bern. Small thing, there are no microscopes in this room anymore. And this is exactly the point. This is because we're moving into the whole digital era. So no more microscopes, away from the microscopes, and going a lot more towards digitization in pathology. So here you see on the left side, we have, this is actually one of our students, but pathologists will be doing exactly the same thing, perhaps with a different kind of microscope. But he has here on the left-hand side, he has these different slides that he puts in under the microscope, and he's evaluating everyone in a single way, in a single fashion. But now what's happening is that we're taking these slides, these microscopic slides, and we're putting them in these nice, beautiful machines, these slide scanners, and we can then produce images that will be, of course, available to us on the computer. So digital pathology is really all about the generation and use of data that can be derived from digital histopathology images, namely from scans of microscopic slides. And there's, of course, a huge added value when it comes to digital pathology, namely in teaching, for very obvious reasons. You can share images across, many, many different students at the same time. You don't need to have these collections of microscopic slides anymore that every student is looking at under the microscope. And in terms of diagnostics and research, we'll go over some of those advantages in the next few slides. So digital pathology is all about creation of this kind of data that we can then use to try to extract really a lot more information from the images that we're seeing now on the screen than we could under the microscope. So there were some revolutions in pathology, at least this is how some of our colleagues have published in histopathology. And basically, the first revolution in pathology occurred in the 1980s, where not only could we look at slides under the microscope, but in fact, we could produce something called immunohistochemistry. Namely, we could look for proteins now under the microscope. So proteins that would be present inside those tissues that could then be used and still be used now for purposes of personalized medicine. So for instance, in breast cancer, every patient with breast cancer who's diagnosed in our institute has a certain series of proteins that are being evaluated in their tissues. And so that was basically the first really big step forward in terms of pathology, which was already some years ago. And then around the 2010, in this era, was of course the next big revolution, the molecular revolution. So now we're not only looking at proteins that can be found on or in the tissues, looking at that under the microscope, but we can grab a huge amount of information from the whole molecular profile of these different cancers using many advanced techniques. So also next generation sequencing is something that is available now very, very frequently. And is again a huge step forward in personalized medicine for patients who, for instance, have different kinds of cancers. And now the third revolution in pathology is not only being able to use the protein information or the molecular information, but also to be able to use the image information to identify features that we otherwise could never have even seen or doubtfully have seen if we didn't have the possibility of analyzing these images digitally on the computer. So we're really now in the third revolution of pathology, where we move away from the microscopic image. Well, we use that microscopic image, but we don't look at it under the microscope anymore. We're trying to go towards digitization of our microscopic slides and pull out a lot of information. However, I must say that going digital in diagnostics is complicated. It doesn't only require a lot of people and resources and money, but it requires so many different interfaces. And this is why going digital takes time, takes a lot of energy, takes a lot of planning. On the one hand, we have slide scanners. So that's basically the essential component of that hardware that we need. So that's those slide scanners that have to be interfaced with a lab information system, which is where, for instance, the pathologists are giving their reports. This has to be interfaced with an image management system that allows us to basically to visualize these slides, to handle these slides, and to have that connection with the LIS so that we know these cases are finished, these cases are new. And something that we learned almost the hard way is that trying to interface third-party software into this whole workflow can be a nightmare, especially if the planning is not well done. because scanners produce different slide formats, and these slide formats are not necessarily compatible with anything that we do downstream. And on the one hand, we're stuck in the sense of we want to be able to have a closed enough system that we can say this is appropriate for accreditation purposes and diagnostic routine. But we also want to be able to use those images to put them into research and to have those developments from research going back into the diagnostic routine after validation. So in our sense here at the University of Bern, we want to have this loop of diagnostics to research, research to diagnostics. It's not always the same needs everywhere, but that's a lot of the time what you'd encounter at a university hospital. So there are so many different components that are involved, and not only hardware and software. I forgot to mention GPUs, CPUs, and storage, since we have an issue with storage. Every slide that we produce digitally is about 2 gigs. So if you imagine we have 450,000 slides per year here at our Institute of Pathology, going digital in one shot would mean 1 petabyte of storage space per year that we would need. So these are humongous considerations that we really need to plan in advance. And of course, we need teams of people. Our MD team is absolutely critical. Our IT team, basically digital pathology is an IT project. So it comes down to that. And also the lab staff that is hugely affected. And of course, depending on your situation, the interactions with the hospital are, of course, very, very important considerations. So why is it so important to go digital? If it's so complicated and requires so much money and so much storage and all of these different considerations, why do we want to even do this? Well, on the one hand, everything we do digital is going to, of course, improve patient safety because we're working in a paperless way. So this is already a huge improvement in terms of diminishing errors. And something that we learned also the hard way during Corona time is that we could now perform remote diagnostics. If we were ready, we could have already done this some months ago and have been ready for this remote diagnostic or possibility to work from home already now. And so in our institute, we had all the different elements in place to do that, but we just didn't have that extra step to actually allow us to really do that from home. But I'm very glad that we took really big advances in this area in the last few months. So you will ask pathologists, and they will say that their diagnostics is not faster doing it on the screen, but there are additional advantages. For instance, pulling out cases that used to be in the archive would mean that somebody would need to go downstairs into the basement, pull out slides, and then these slides could then be given to our pathologists who would then share their diagnoses with our clinical colleagues. But now it would be very easy to go and search for slides. You go into your computer and there you have it. So anything to do with second opinions, instead of shipping slides around the world, you would then be able just to send a link to your colleagues. And we're doing that right now. We're also sharing between, for instance, Lausanne and Bern, almost on a daily basis, we're sharing slides for basically to have a look at for second opinions and so on. But one, of course, major advantage is also in the sense of personalized medicine. If we have this image on the screen, we can now perform more accurate measurements. We can now look at biomarkers, and I'll show you this in the next couple of slides, how we can evaluate biomarkers, ideally in a more objective way. And this will lead, hopefully, to more reproducible results and also better documentation, because what we can see, we can put directly into the reports. And this just generally will improve the quality of the service in pathology. So some of the basic, or if we could call it like this, basic image analysis tasks are, for instance, biomarker quantification, which I'll show you, tumor detection, and some other features like tumor grading and tumor subtyping. These three are based on deep learning. Biomarker quantification does not necessarily have to be a deep learning step. And there's now been a lot of publications coming out in the last, let's say, 5 years that are really quite strikingly advancing this whole field. So I'm going to show you a couple of examples. So in terms of biomarker quantification, this is something that our pathologists are going to see daily in the institute. So as I mentioned, every breast cancer that comes in will be stained for a certain series of markers. For instance, here, this is not a breast cancer biopsy, but it shows you different kinds of staining. So here on the left-hand side, actually, we have the same staining. It's a staining for HER2. And I think that probably everybody who's a bit familiar with immunohistochemistry will obviously see that whatever is brown is considered positive. So this tissue specimen on the left side would be a positive specimen. The tissue on the right would certainly be a negative specimen. But this tissue in the middle is sort of like, could be positive, could be negative, sort of depends on what you see. So that's where image analysis can really start to help. Here we have a daily problem. It's for a protein called Ki67. This is a proliferation marker, and these are breast cancer biopsies that you're seeing here on the screen. And this Ki67 analysis will give us information, or give our pathologist information, who can then transfer that information to the oncologist in order to better make decisions about whether certain patients could go or should be included for chemotherapy or not. And in the end, what is important to know are these patients positive or negative for Ki67? And what is the percentage of Ki67 positive tumor that we're seeing in this particular biopsy? But this problem of positivity and cutoff is a very tricky one, because it has been shown over and over again, also using clinical trials, and also published very nicely by colleagues in Zurich, that the inter-observer agreement in such a declaration of positive or negative case is very low, especially in certain types of breast cancers. So how can we solve this potential problem? And of course, one possibility is through image analysis. So this is a tool that we developed ourselves in-house, and it basically collects all of the breast cancer biopsies that are being scanned by us. So we do that in a daily way. So every breast cancer biopsy that comes to the Institute is scanned and will go basically into this project. And for the moment, because we wanted to gain our own experiences with image analysis before, let's say, buying any commercial software, or before really going into, validation of our own, algorithms ourselves, we sort of put this in a research context to see, for all of those biopsies that are coming in, could we create our own classifiers? So this is the first example of something that we did. So this is a breast cancer biopsy stained for Ki67. That's what you see as these like nice dark nuclei. And here a pathologist is making the region of interest around that area that they think is an important area. And then they will run our algorithm. And our algorithm here first leads to a detection of the tissue, then detection of the tumor within that area that is the region of interest, and separates out the tumor from the stroma. And then we'll count the Ki67 positive cells only within the area of the tumor, and we'll then produce here, for instance, this percentage of positivity, which we can put then back into our database. And we're growing this database at the moment, so we have more than 100 cases for whom we have this. And now we've made some really nice studies comparing commercially available software to a ground truth, namely a poor guy counted literally every single cell in this region of interest to make sure that what we were creating was actually, making sense. So that's an example of real life situation that we have at the Institute right now. Something that now goes beyond that is, for instance, going away from biomarkers, now going into a different task, for instance, tumor detection. And this becomes very interesting because in this case, we don't need any sort of maybe special stainings or anything like this. We can use the standard H&E image that is produced everywhere in this world as the basis for everything that is diagnosed pathologically. So here on the H&E slide, this might not be the earliest example of tumor detection, but it certainly is one of the more famous ones. Because this was a challenge that was put out called Chameleon 16 and Chameleon 17 that looked for metastases in the sentinel lymph nodes of patients with breast cancer. And so this was a challenge. And in the end, lucky for the rest of the community, they published all of these different slides that could then be used for anybody else to train their algorithms on, sort of use that as a basis for future tumor detection or lymph node detection algorithms. So we are also doing the same thing. This is a project by one of our students who is interested in developing a tool for metastasis detection in lymph nodes. And this is a real life colorectal cancer case that you're seeing here. So one thing, of course, is the sentinel lymph node, whereas the one lymph node is being looked at. But here, in a colon cancer case, you might get a huge amount of slides that you need to evaluate, or that pathologists will need to go through every single lymph node to look for even the slightest amount of cancer in any of these nodes. So you can imagine what kind of a task this is. On the left side, these are the tumor slides and other slides. Actually, these are not only tumor, different slides that come together with that tumor, with that cancer that has been resected, for instance. And many of those lymph nodes are also taken out along with that cancer. So you receive here basically a very large specimen containing the cancer, and of course containing that surrounding tissue. So our pathologist's job, first and foremost, is to be able to pick out all of the lymph nodes that are around that tumor. And after these slides have been produced, to go through every single one of these and look for the slightest cancer cell. So not only a mass, but even down to something like an isolated tumor cell. And you can imagine, this is really labor intensive. At the end, this particular patient had 88 lymph nodes examined and 0 contained any tumor. So what we would like to do is produce a tool that we can put into our routine eventually to actually screen all of these as sort of a first look. So there's this concept now of first look, second look, and sort of a first look screening to show, yes, there is cancer, for instance, here and here, so that our pathologist can confirm this. And of course, this will not be used as a tool that you rely on for making the diagnostic without even looking at the slides, but will ideally be something very complementary and sort of facilitate this task. So where are we right now in this project? Well, we have here, for instance, these are lymph nodes that are on the slide. So in the first step, we have very, very successfully already segmented these lymph nodes. And in the second step, we're doing the metastasis detection, which this is the ground truth annotated by our pathologist. And this is actually what is looking like right now using our algorithm. We have a couple of false positives here. And if you're interested, I can tell you what that is then later. And ideally with this tool, we'll be able to implement that into our image management system and sort of have that, production of, or that computational part being performed in the background as the pathologists are already receiving their images. So tumor detection and grading as a second look has also been published recently in prostate cancer. And this is quite a fantastic study that was performed to show how, again, an algorithm can detect these prostate cancer within these biopsies. So again, here you have prostate cancer corneal biopsies, and not only can detect the cancer, but can also grade it. There is this grading scheme called the Gleason grading in patients with prostate cancer, and the higher the grade, the worse the differentiation of these of this tumor is. So in this case, this is actually a commercially available software that can be bought and is now used as a second look. Namely, pathologists will first make their diagnosis, and then the software is run over it to say, is there a match between the diagnosis saying yes, no cancer, and also for the Gleason grading. So this has actually become very advanced. But isn't there more? I mean, looking at this image, this is so informative for anybody who understands or what these cells are telling them. This is hugely informative. I mean, we're not only seeing here, these are cancer cells, and this cancer is really invading into that stroma here, but we can see how these cancer cells have popped off of the ends of this tumor. This is something we call tumor budding. And we can see how there's this huge infiltration of immune cells battling against that cancer. And we can see how vulnerable these vessels are, and even invasion of cancer, perhaps here inside these vessels. So this is an incredible composition of different cell types performing different functions in such a spatially arranged way that, there's so much information that we could be deriving that is obviously not reported in daily diagnostic routine. We have a series of elements reported for colon cancer, but nothing that can really sort of, grab that information here. So the more advanced image analysis tasks are now going to be looking at, of course, new prognostic factors or survival factors that we can look at or gain from these images. Either ones that we know are there that could potentially be excellent prognostic factors, but also ones that are new. We'd also be using this image analysis task to look for response prediction. I'll give you one example of somebody who's published recently on this, and I think it's a really nice study. And the third thing that we're looking at is, can we get to the bottom of a genotype of these cancers just by looking at an image, that we wouldn't have to be doing this expensive RNA sequencing that is, of course, a phenomenal tool for precision medicine. But what can we learn from the image about those molecular alterations and how they are, being visualized from the image? So these are fascinating points. The first is a study here by our student Linda. So she's working on, as I was mentioning, these things called tumor buds. So these are these cells that have kind of popped off of the ends of the tumors and are obviously responsible for causing metastases in patients with not only colorectal cancer, but with many different tumor types. And she's using a graph-based approach to try to understand how the spatial arrangement between tumor buds and the different lymphocytes, and these are CD8 positive T cells, which are, as if you remember, sort of those cells battling that cancer that I showed you on the other slide, even though that was an H&E. So she has access now to these double stained images, so to cytokeratin staining, which is identifying our tumor cells here, these beautiful buds, and of course these CD8 positive lymphocytes, which are in pink. And ideally, by being able really to have these, to understand that spatial arrangement, that sort of impression using her graph-based method, we can then also start to understand whether that can be related to survival. So this is really interesting work. Something that is also being done, and this is a part of our really nice collaboration between Jean-Philippe's group and ours here. This is work by our student Christian, who is also gaining a lot of inspiration from looking at the whole slide images and trying to understand new prognostic factors from these. So by, first of all, developing new methods for segmentation of these different tissues. So for instance, here we have the segmentation of the tumor and of the stroma, and there is something that we call the tumor stroma ratio. It's not very original, but this has been shown to be a very interesting prognostic factor. And if we are able to do this in an elegant and automated way across all of our different tissue samples that we have here, we could potentially really show that the tumor stroma ratio is a highly valuable prognostic factor. And it's already encompassing a lot of different elements. Tumor and stroma is sort of vague, but we can take it one step further even, and not only look at tumor and stroma, but as he has done here by solving this sort of multi-class detection problem, really extrapolate that sort of tissue or tumor stroma interaction to looking at many more different classes and seeing how their interactions can really help us to identify new prognostic factors for colorectal cancer, and of course other tissue types as well. So going on to the idea of the genotype prediction of phenotype problem. Can it be done? So can we look at an image and say, this cancer has this kind of a genotype or these kinds of mutations inside the tumor? Well, let's take the example of microsatellite instability. This is a fancy word to describe tumors that have defective DNA repair systems. So normally, the DNA is very well repaired. There are excellent systems for repairing any kind of mismatches between incorrect bases in DNA, and our regular enzymatic system will come and cut off these bad regions and basically create beautiful strands of DNA, which will then replicate in a proper way. But when that doesn't happen, there is a defect in the DNA mismatch repair machinery, and this can lead to certain problems. So those kinds of tumors that have that are hypermutated, so they have a bunch of mutations because the DNA is not being corrected. And they also are very typically hypermethylated. Doesn't really matter if you don't know what this really means, but this is like, probably you know that inside the DNA you get mutations that can occur, but you can also get methods that sort of screw up a little bit how your DNA is then being then further transcribed And this is by something they call epigenetics. So these are like molecules or for instance, methyl groups in this case, that are being added onto the DNA, which shut down transcription of that particular gene of interest. Anyways, the importance of microsatellite instability is at least threefold in colorectal cancer. First, diagnostically. Patients who have microsatellite unstable tumors are very often those that have a familial heritage. So they have a familial predisposition for getting colorectal cancers. But there are also patients who have these sporadic colorectal cancers, and they have then quite some consequence. So MSI tumors will typically be very good prognostically, but they will also respond very badly to chemotherapies that have a particular chemotherapy inside, so 5-FU, but they will respond extremely well to immunotherapies. So that's why we are very interested in microsatellite instability. So can it be done? Can we predict microsatellite instability from an image? Well, it's been done before. This was in 2007, actually, in the group of Jeremy Jans, who happened to be my PhD supervisor in Montreal many years ago. They developed a sort of microsatellite path score, and it was almost 100% accurate. Trained pathologists can look at an image and say, this must be an MSI cancer. And how do they do it? Because they consider the age of the patient on the one hand, they consider where that particular tumor is, if it's on the right side of the colon or left side and so on. But they also look at certain features, and they can say in one glance, this seems to be a microsatellite unstable tumor because they've identified certain features that are quite predictive of that. So it is possible to do that. And if we can do that by eye, imagine if we could even use our computational approaches here to pull out even more information. And recently this has been done. So Cather and colleagues in gastroenterology published what they call as a clinical grade algorithm for microsatellite instability detection. And they've, of course, done this using international cohorts, thousands of patients. They have achieved excellent AUCs. So classification was very, very high in the external validation cohort. And they even have here these nice probability maps showing more or less, the region where the decision for having an MSI tumor or not was actually made. So in some cases, it's not so obvious as here. In some cases, it's very obvious. Everything yellow here would be a tumor. Well, this is highly predictive of being an MSI tumor. But the problem is, and this is again the most interesting thing, is even though the prediction is working so super well, what features are leading, of course, to the prediction of MSI? What features? Well, we know for sure that one feature that seems to be of very important, very interesting, or very important for the classification of MSI tumors is mucin. And mucin is something that we see also, and this is a project by our postdoc Yao in the group, who has produced an algorithm that allows us to detect and quantify mucin within colorectal cancers. So why is it producing mucin? Well, these are exactly these kinds of cancers that will be producing mucin, and this happens in up to 12, maybe 15% of all cases. But we do know that mucin is only one feature of the MSI subtype. We also found that mucin production can be very, very prominent in patients who have a different kind of molecular subtype, the CMS3, and of course CMS4, but the CMS3 are particularly interesting. And what I really like about our project here is that we not only could identify that mucin production is not only a feature of MSI, but also of this other molecular subtype, but when we go back to actually analyze the genes expressed in these tumors, we then also see that mucin-producing genes are highly overexpressed in those subtypes. So again, we go from the image to the molecular subtype, and now we can even validate that using our single genes. So I really like this example. This is something that's not from our group, but I just wanted to show this because this is a very, very new publication showing basically for the first time that the ERBB2, which is a very important molecule being analyzed for breast cancers, that using computational methods, not only can this study by Johan Lundin's group and by Dmitry Bichov, they have shown that you can identify these tumors from the H&E slides. So you can predict this particular genetic aberration from the H&E slides. But even in the case where these mutations are negative, so there are no mutations, using their images, they could still stratify patients into different prognostic subtypes. On the one hand, they have a beautiful prediction of response to this anti-HER2 therapy, so this one here, and they can show that in their cases where they've identified this ERBB2 mutation, that these patients are responding very well in contrast when they receive this therapy, in contrast to those who do not. But even in the cases that are negative, they can still stratify these patients into different prognostic subgroups. This is a very interesting paper. However, even in that case, what are the areas that are leading us to the prediction of certain classifications in terms of this molecular background? And now the whole question becomes super interesting. And how can we bring that back to the biology of the tumors? And this allows us now the entry into a whole new domain for pathology, which is combining what we are learning through these computational approaches that we can then apply back to understand biology. And one way that I foresee that this is going to happen is by using such methods, and even in a more higher dimensional state. So this is a 6-plex. So this, you can look for 6 different proteins at the same time, basically on the same image. And this will eventually be carried out for 10 proteins, 20 proteins, 30 proteins. And we can combine morphology with this information coming to us from biology. So how can we do that in a way that's also quite cost-saving? There are these tools that can be used for that. And these are called tissue microarrays. So what you're seeing here in this very big picture is maybe only something this big, okay? So one of these blocks is only something this big. But tissue microarrays can not only produce a huge amount of images, but they will allow us to inform on the biology of the patients or of the cancer samples that we're looking at in a way that will be very effective. So what you're seeing in this image is actually a paraffin block, and inside each of these, so what you see here as every single spot is a piece of cancer that has come from one or more patients. So here we have, obviously, from more patients. So we have a spot here, which is a core that has been taken out from a single area within a cancer and placed inside this TMA. And if we do it again and again and again and again, you basically will end up here with a, let's say, matrix, something that looks like a matrix, whereby you might have the first 6 or 8 spots being from the same patient, and then from another patient, then from another patient, and so on. So you can have an analysis of cancers from different patients that you can concentrate together into only one such block. And there are huge advantages for doing that. What we do in our lab is we use digital pathology as a basis for constructing these tissue microarrays. So here on the left side, you see an image. And of course, this is a colon cancer that is very heterogeneous in terms of its morphology and its tissue composition, etc. And there are different areas that we're interested in. The center of the tumor is different from the invasion front of the tumor, which is different from those areas of budding that I already previously discussed. So we can actually annotate this image and then align an image together with that block from which it came from. So this is the actual cancer piece. And then, of course, there's that cut that's made, and this is that cut that normally goes under the microscope. But we can annotate these images and say, we're interested in looking at that. And we want to do that across 100 patients. So this is what we do. We end up here coring out these different regions that have been annotated after this alignment between the block and the slide has been made. And in the end, those regions are punched out. They go into such a block as you saw on the previous slide. And then once that's cut, it looks like this. So this is an H&E staining of a TMA that was produced from hundreds of different patients and looking at their different colorectal cancers. So this is how our facility looks like at the moment. We have basically these 2 machines. that are producing these tissue microarrays for us. And we do this in a very sort of large way. So many people now, we have the chance to collaborate with so many different people from around the world. And this always is very interesting for us because we get to see what sort of, what are the next trends that are coming up for researchers, and what are they interested in. So our MGTMA collective at the moment is more than 800 different blocks. We have a bunch of different tissue types that we are evaluating with these tissue microarrays so that we've produced. We've punched more than 150,000 punches directly into those blocks. So you can imagine that if you punched so many tissue spots into those blocks and you cut them and you scan all of those tissue spots that you've ever done, you could basically construct a huge archive of digital images. So what do we use these tissue microarrays for? Well, we use them for precision medicine purposes. We try to understand what proteins are involved in these different cancer spots that would allow a prediction of prognosis or a prediction of response. We study tumor heterogeneity using these different methods. We study rare diseases where material is maybe very precious. So we don't necessarily want to keep cutting down these cancer specimens. Maybe these are pre and post immunotherapy specimens, and these are very, very precious. This is very precious material. We could construct, for instance, a tissue microarray, so we're only evaluating single areas. And of course, if we're interested to study targeted histological areas, then, for instance, our budding, which was actually the reason why we even went into the whole TMA field, we could study this because we have digital pathology now. We use this also for animal studies and to establish new methods. And here are some of the examples. So just to give you the visual of how it looks like, these are, for instance, double stains of, I think in this case, the PDL1, and perhaps here this might have been a cytokeratin, I'm not so sure, or anti-CD8, yeah, anti-CD8 and anti-PD1, okay. Here we have a fluorescence, so double fluorescence that was also performed. On the other hand, here we have an mRNA in situ hybridization or standard DNA fish, and even just, standard immunohistochemistry. But in this case, we can really, study, for instance, primary tumors and very special kinds of tissues like brain metastases of patients who have breast cancers. And there are certain trends in the tissue microarray world, and one of them I was mentioning earlier is the multiplexing, where we see this as going. This is going to certainly be something that will be extremely valuable in the next times. So multiplexing is, of course, ways of looking at not only one protein or two proteins, but we want to continue with 3, 4, 6, 10, 20 proteins. And of course, computationally, this becomes so insane. It produces such a huge amount of data, but this is what it's going to be about. Multiplexing can be very expensive. So ideally, you want to try to concentrate your tissue to a smaller area if possible. If you want to scan such an image, scanning will take forever. The more, of course, number of proteins you have as well. And the analysis can be very, very time consuming. So this is really at the beginning of this whole new dimension. Well, we've tried this one time. So we did produce a tissue microarray together with our colleagues from Stanford. So from Gary Nolan's lab, they were developing a new technique called codex. And the question was about looking for different kinds of aggregates of lymphocytes within, or lymphoid follicles or lymphoid aggregates in colorectal cancers. So the lymphocytes can be distributed in different ways in the colon cancer. On the one hand, you get these sort of like patches or these really nice follicles. But on the other hand, you might get something that covers more or less diffusely the border of the different tumors. So the question for them was, is there a difference between those kind of more organized follicles and that more, let's say, diffuse infiltration of lymphocytes at the invasion front? And this is a problem that we could only solve by really using digital pathology to annotate those specific regions of interest and then study those. So this is what we did. We punched out those different areas. And then what happened was that so this is not only from our colorectal cancers, but this is the technique that they use, codex, So where they label 3 antibodies at a time, so they use 3 antibodies at a time, they label and strip them. So by doing this, they can basically keep adding on different proteins that they can then visualize by modifying these colors. And so here we go. We're looking here only at a handful on this particular image because more than this is simply difficult to even imagine, but that's what it is. And in the end, what they look at is by using these sort of sliding window approach, and they sort of slide this over this whole TMA punch where that core was taken from, that includes all of these lymphocytes, and they're able then to come up with, you know, different clusters and neighborhoods. So what is the content, for instance, of this neighborhood, and how are these neighborhoods connected to each other? And then they end up analyzing this whole complexity. So imagine now going back to the standard H&E slide. So you can, I'm sure you can see how powerful this combination will be. We're also able to use our technique for tissue microarraying, not only to punch out cores and put them into that TMA, but we can punch them out and put them into little tubes. And by doing this, and also, of course, creating tissue microarrays at the same time, we get a tool that will allow us now to combine the morphology that we see from the TMA to molecular analysis that's being done downstream. Because using this material, it's good enough to do some RNA-seq, some targeted RNA-seq, and to some degree, some whole exome sequencing. And if you look at this publication by Mark Rubin's group very recently, they're able to use this information to study the clonal evolution of prostate cancers from benign tumors to the prostate cancer lesions themselves, to then in a very special region, brain metastases. And they've done this Swiss-wide study to analyze this using this material. So again, combining methodologies is going to be something I think will be the next forefront in digital pathology. And as I mentioned, imagine we've produced many of these TMAs, and every time we've ever cut a TMA, stained a TMA, we've scanned it. So in essence, almost like by accident, because we've been doing this for 10 years, and we thought, the real reason why we actually made the scans, to be completely honest, at the beginning, was that we didn't really trust our pathologists. You give them a slide and they're going to lose it somewhere. And we know that they're going to come back to us and say, oh, do you happen to have that slide? No, we gave it to you. And then 5 years later, it will be under the microscope or something like this. So before we handed out any TMAs to anybody, we scanned them and inadvertently created a gigantic repository of images that we can now, of course, use for training of algorithms. So we have more than 1 million of such tissue punches. And the beauty of what we have is that our pathologists were working together with our clinical colleagues so nicely that for almost every spot, we have full follow-up and treatment data available. So our images come from 3 sources. We have this beautiful, actually annotated, high-quality annotated labeled data from pathologists. We have the content inside each of these spots, and we, of course, have the TMA spots then afterwards when they're cut down. The thing is that when you cut down these TMAs, you might be changing the content inside those punches. So this is something that Yao, our postdoc, and the group we have put together, which is a way to reclassify each one of our tissue microarray spots. And to train such an algorithm, we actually used our own archive for this, which contained more than 15,000 different H&E spots of colorectal cancers. This is pretty much almost my last slide. One of the big challenges that we have now, and you can imagine, these are scans from just my colorectal cancer TMA. This is more than 300,000 spots. And each one of these has to be then related back to all of these different parameters coming from the patient. And how can we manage this data? This is something that by far is the biggest challenge. So where do we stand in terms of clinical applications? Well, we're getting there. We're getting there. There is a lot of research going on. Now there are also guidelines that are being set up for how to report AI-related studies for digital pathology. I suppose this is also heavily based on previous experiences coming from your field. So in terms of clinical application, there is maybe one, this might be now 2 validated, let's say, algorithms for clinical use. More than that, we do not have. But there are huge benefits for accessing data, sharing data, being able to use all these algorithms coming from all of you to apply that to digital images. But we also have a lot of issues to contend with. We need tons of storage. And this is something that we don't, we're just learning to deal with now, or GPUs, our HPCs. All of this stuff is brand new for us in digital pathology. And I think that certainly combining modalities is going to be the biggest, most powerful thing we can do, and ideally will help us to go back and understand biology. What are we learning from these images, and how do we understand that from the biological point of view? And with that, I hope I'm still on time. I would like to thank my team here, my digital pathology team in Bern on the research side, on the diagnostic side, and of course, all of our collaborators whom we work very closely with, especially Jean-Philippe. It's been a really interesting last couple of years, and I look so forward to continuing to work together. And I should mention that there is a new Center for AI in Medicine here at the University of Bern, led by, I think, somebody you are very familiar with. He's the director of this new center. And there's also a master program that is coming out from there called the Master for AI in Medicine. So just to have said it in case you have some students who might be interested. And thank you very much for your attention, and I hope it was interesting for you, and I'm happy to take questions.
Okay. Thank you so much, Inti, for the great talk. So time for questions. I'm sure we have plenty of questions from the audience. I would like you to raise your hand if you want to ask a question, so that we go in right order. And for you to have some time to think about the question, maybe I can start with one question. Inti, can you just elaborate a bit on whether, if you ignore the question of the amount of data, whether the third dimension would add something in this analysis? Would it be interesting to have either multiple slices analyzed jointly, or going to another modality where you would have the third dimension?
So that is certainly what is completely missing in pathology. We are only seeing everything in 2 dimensions. And there would be huge benefits to getting the third dimension, not only from a quality control point of view, but for being able to communicate, to improve the diagnoses of patients for whom we also have radiological images. I mean, these are our radiologist colleagues. Oftentimes, the pathologists and radiologists may not always agree on what they're seeing. And I think from that point of view, to combine radiology is like pathology viewed in a different way. And to be able to combine those 2 modalities would be an incredible breakthrough, frankly. I think that I've seen a couple of chats on LinkedIn that this would be, people are intending to do this, that they think it's a fantastic idea. And certainly this would propel us probably into the 4th revolution of pathology, is to bring in a third dimension. And what's been done so far, I mean, there are a lot of different methods for looking at the third dimension. One thing that we've originally tried here, but this was years ago, is to actually cut down that whole specimen and then try to rebuild it by using, for instance, some immunomarkers or something like this to try to trace, for instance, where the vessels are inside, so we can align the different images on top of each other. But it's difficult, very tedious, and I don't think at the end it's not that kind of information we want to have. So I think combining certainly, something from our radiology images together with pathology would inform hugely diagnostically and research as well.
Thank you very much. So maybe a question from you, Mary. Can I ask you to introduce yourself very quickly before asking your question, so that we
sure.
know you?
Hello, Inti. I'm Mary. I'm one of the section heads of the CIBM at UNIL SCHUF, as well on the signal processing part. And I really enjoyed your presentation. It's really amazing. I'm not familiar, so I discovered a lot of things. And one of my first questions, you have tons of data, tons of images. And one of the things that it happens when we want to mix these images in the, so I'm from MRI, is that we need to deal with image artifacts and harmonization because different centers, different scanners and so on. So may I just ask you, is this happening as well in your images? Do you need for such image artifact removals, or are the images from different devices comparable out of the box, or you need to proceed?
So happy for this question. And I put this in hoping maybe somebody would ask.
Okay.
So here, this is exactly what you are talking about. This is the same image scanned with 4 different scanners. And you can clearly see that there are differences. You know, some have an excellent contrast, others less. And how does this affect our algorithms downstream? So there is, you know, ideally, we try to gather slides from different institutes named under different conditions so that we can expose the algorithms that we're developing to all these different varieties. Because we know that the algorithms that we'd like to develop should be generalizable to everybody, with hopefully as little pre-processing as necessary. In many of our studies, we have to normalize, of course, for the differences across the color variations and so on. So that is being done. But one of the ideas, especially from this, for instance, I showed something about this commercially available software for prostate cancer detection. They validated this on 6 million slides coming from everywhere, to the point where none of this, hopefully, normalization needs to even take place. So it's so robust. And it will basically absorb all of these differences into the algorithm, that none of this will happen. But you're right that this is a daily occurrence. And even if we change the software version of the scanner on which we are making these images, it might be different. It's the same producer, same scanner, same everything. We might have 2 of the exact same instruments even in the institute, and they will produce different images. So to be able to control for that is something that is a very important question. Another problem that we have is real artifacts. Like, you know, artifacts in terms of what we would call artifacts would be like, okay, out of focus images, or images where we have folds. That's something that's really annoying, because if you have a folded tissue piece here, for instance, that's like, it will create, you know, it's, we don't have any folds on this because, of course, it's the perfect image to show you. But it would create something that would either, it's a thickness, which leads, again, to out of focus, or to it being detected as something it shouldn't be. So this is a problem, and we are actually working, there's a master student of ours who's working on the QC issue. So in other words, before these slides go on, ideally to pathologists later, that those QC issues will have been identified and either been sent back to the lab for recutting even, so depending on the issue, either rescanning or going really back to the block and recutting, again, from zero to try to eliminate those artifacts. But it's a definite problem, yeah.
Thank you.
Okay, thank you. Robert, question from you.
Yeah, sure. It's Robert Boré from EMPA. I think Inti we met before. Gabi Braga, we are the guys who are doing the imaging. So thank you very much for your very interesting talk. And I would like to just shortly come back to the question of Jean-Philippe, which actually I was just about to ask. I didn't find the raise hand button quick enough. So the third dimension. So as an X-ray specialist, I can just confirm we are actually looking exactly in this direction. So basically micro CT or phase contrast micro CT of full biopsy block. So it was nice to hear from you that could be the 4th generation. So if I were to say to you that you don't need to, probably don't need even to stain, and you can work on full biopsy blocks and 3D, looking at vascularization, for example, that you mentioned, I understand this would be something really revolutionary for.
I believe. I believe.
Okay, so
exactly, and I mean, I could even imagine it being, you know, something like a tool that not only for comparison of diagnostics between, let's say, radiology and pathology to really, you know, argue about the diagnosis for the patient, but also even just as simple as you have a lymph node. Where do you cut it? We cut it like that. We split it in 2, make a cut, look if there's cancer. But if there's cancer on the edge, you're not seeing it. So ideally, if we could somehow have it that we get we get that whole third dimension, we get the volume of it, and we could see contrasts in there that show us, look here. You know, then our pathologists know, path, there it is, it's positive. And I'm so wondering, you know, how many lymph node metastases we are missing by doing exactly this, transforming our 3 dimensions into 2 and then missing it. You know, this is a study that probably nobody would really, I don't know if they'll fund it, but because who knows what the outcome will be, but this would be extremely interesting.
Okay. It's very nice confirmation that we are thinking in the right direction. And maybe just another question, maybe look out in, so you mentioned one of your students looking and using graph theory for the connection between different cells within the cancer. So do you happen to use, or do you know some other groups that are looking at the network science method? So there is a very strongly emerging network science when they are looking at the basic structures, morphology with using graph theory of networks, where is the most vulnerable points of network and things like that. Do you happen to look into something like this, or you are aware of someone?
So we are not, but you may like to reach Andreas Fischer. He's also our expert in graphs working with us from the University of Fribourg. But of course, there are many experts, I'm sure, on this call here. But he's particularly somebody with whom we're looking at that from a histological point of view. And he actually comes from a completely different field outside of medicine. So from document analysis and things like this. So maybe another perspective on that theory. But ourselves, we're not going so deeply into it. We're really here. We're doing a lot of the more applied, more clinical work, I would say. And Jean-Philippe takes care of the theoretical aspects in particular.
Okay, thank you,
Robert. Olivia, you have a question? And if you can introduce yourself first.
Yes, hello everyone. So I'm Olivia from the positron emission tomography section here in Geneva at the UNIGE HUG section from CIBM. Thank you very much for this very impressive presentation. This is really exciting work. I wish I had this kind of tool when I was doing my studies instead of taking hours in front of the computer to analyze slice by slice images. So this is really exciting. One question I have is about the kind of errors you can get from your tool on top of artifact problems. Do you have other errors from this analyzing tool?
So one thing that does happen is, for instance, detection of stain artifacts happen for us quite a lot. So for instance, something that might be detected as, these are things that are quite particular, but let's say the outside of that slide that's being stained could sometimes have a rim of color on it, which is like a staining artifact from the machine, basically. And that can often be viewed as or interpreted as something else. And then that's kind of calculated inside our positive result. And that's annoying. We have there is a software that we are also evaluating at the moment, which is our first what we call zero-click algorithm. So it's basically like from the barcode of that slide, that the particular algorithm is launched. So once that slide is scanned, it goes into a particular folder, and then an algorithm is launched, and then it will identify certain regions of interest, and then it will calculate the number of cells that's positive for something. And then in the end, it's shown to our pathologists, and then they can, of course, decide, do they accept that these results are really true, or do they want to eliminate it? And very often, that sort of region of interest to eliminate is due to something like this, where there's like a border of strange staining that needs to be eliminated, and then a recalculation needs to be made. And this is going to become very tricky for us when we implement this into our real life system, this interaction between pathologist and outcome of an image analysis. And how can we deal with this? Might be a little bit tricky. But those are the kinds of things we run into, is a lot of on-slide issues, actually. Or scanning related issues. If the scanner is not, this is another weird situation. we had an issue with the tiling of like the stitching and tiling of the image being produced by our scanners. And it produced gray boxes. So it was like differences in contrast. And you can imagine that for our tissue microarrays, these gray boxes were being picked up as spots. So this is an issue we had for months. And it was a big fight between us and the company, who were like not believing us that this was a big problem, but it was. You know, to think that further, you know, that not only is the image being, but further than that, because actually the requirements for pathologists' diagnosis and then algorithms being put on images is different. It's a higher quality necessary, of course, for the algorithms. Yeah.
Okay.
Thank you very much. I have a second question because I'm in the positron emission tomography section. So my question is, have you ever did this kind of work with autoradiography slices to maybe help clinicians to refine their analyze of the different tumors?
So we have not. There was somebody in our institute, Mr. Royby, who was doing this quite some time ago, but this is not something that's being done now, I don't think. No, not here. Not certainly by us here.
Okay, thank you.
Thank you, Olivia. Matthias, questions from you?
Thank you very much. I almost forgot my question. I think it relates to, my name is Matthias Stuber. I'm the section head of the CIBM MRI section at the CHUV. And it relates to the question of Jean-Phi already. I mean, he mentioned 3D, and you are looking at three-dimensional structures, and I think there is no way around going 3D also in pathology. Now, of course, Jean-Phi already mentioned it. If you forget the size of the data, and I think that's a valid assumption to make, because if you think back about memory size and how it has evolved and Moore's law, et cetera, we probably shouldn't worry too much about that. But nevertheless, there is a contemporary challenge that's related to this. And I wonder, you mentioned MRI or other imaging modalities, could also be phase X-ray for the sake of this meeting here. Would transfer learning be something that would help augment this query for three-dimensional information?
So you mean basically taking what we've learned from other modalities to apply them to pathology?
Yeah.
Certainly. I mean, I don't really know how to answer the question in the sense of I don't know exactly how you are defining transfer learning in this context, but certainly I would say being able to combine what we are seeing together with different modalities is certainly the way to go. And not only, we're okay, the 3D component of it, but also being able to use what we have from our histological slides, and then being able to add on our molecular information from there, to be able to add on the protein information from there. And that is going to really complete the whole story for sure. And what we can extrapolate from 2 dimensions into a 3rd dimension, or just the information that we can grab from the 2nd dimension already now, is going to be, I think, hugely valuable. So I'm not sure if I answer your question this way, but yeah.
Well, I think we would also need Jean-Phi's input on that, what he thinks about this, because he knows.
I agree that we have to find a way to leverage the incredible amount of data that you already have, where the features are actually extracted already. You know what are the features, but when going to 3D, you can reuse that and basically learn the 3D structure while leveraging what you have done in 2D. I think that's probably what Matthias has in mind.
Oh yeah.
On the one hand, for the microscopy, for the pathology images. Then if you are talking multimodality, and I agree with you, here also you have to basically see what you can learn, because this amount of data that you have, that your community has been processing for years needs to be used, and we cannot start from scratch. No way.
You know, what is quite a pity is that a lot of these images that we have are just not available. I mean, if you try to, you know, if this chameleon data set of lymph nodes is one of the rare cases that people are actually publicly making available for us to be able to use as well. So now they, you know, to be able to share the data as well, so that we can not always have to start from zero, you know, would be tremendously important.
Exactly. And one of the good things in this direction is that, correct me if I'm wrong, but most of the tissue that you're considering for cancer are kind of isotropic. I mean, they don't have real privileged direction or privileged orientation, as opposed to the white matter in the brain, where you have fibers that have specific orientations. So I guess what you are doing in 2D would extend to 3D without adding additional features in most of the cases, at least, if you see what
I mean, in terms of orientation from the pathologist's point of view, it's about how you orient that tissue sample inside that cassette when you cut it, so we can get the most information.
Indeed. Okay, I think we will stop here. Please join me to thank again Inti for the great talk. Thank you very much, Inti.
Thank you.
And Pinat, back to you.
Thank you very much once again on behalf of everyone.